Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
For research, plants are frequently grown under stable lighting, which does not reflect natural conditions. In a series of experiments with changing light conditions, simulating the natural interplay of light and shadow, researchers from the Max Planck Institute of Molecular Plant Physiology in Potsdam-Golm, Germany, and the College of Natural Science at Michigan State University, USA, reveal the important. Plants perform photosynthesis to grow. In this process they use energy from sunlight, release oxygen, and produce carbohydrates, which are the basic food resource for all humans and almost all animals on Earth. Under natural conditions, light availability can change rapidly in a very short time. One of the main reasons are clouds, which provide light and shadow as they pass in front of the sun. Plant leaves and branches can also temporarily provide shade when they are moved by the wind. Plants cannot move from shade to sun when light is limited, and conversely, cannot evade from sun to shade when exposed to too much sunlight. They have to respond to changing light conditions in other ways. Just like for humans, too much sunlight is harmful to plants. In particular, a rapid change between faint and intense light is problematic. Like the retina in our eyes, plants use molecules in their leaves to capture light particles. When light is low, these light traps are very efficient at catching as much of the low light as possible. If light conditions suddenly change, too much light energy might reach the plant. This energy can overload or damage the sensitive photosynthetic apparatus inside the plant cells. Accordingly, plants have to constantly adapt their photosynthetic activity to their environmental conditions in order to obtain maximum light yield on the one hand, but avoid being harmed by too much light on the other hand. To date, plants in greenhouses and laboratories are grown almost exclusively under stable and uniform light conditions. Therefore, our understanding of how adaptation to changing light conditions works is very limited. In the worst case, this can lead to plants that are growing well in laboratories and greenhouses but suddenly perform much worse than expected when cultured in the field. Regulation of photosynthesis under changing light conditions. The researchers around Ute Armbruster from the Max Planck Institute of Molecular Plant Physiology in Potsdam-Golm and David Kramer from the College of Natural Science at Michigan State University, USA, examined the model plant Arabidopsis. Plants were grown under a wide variety of conditions including static, fluctuating and natural light. The study focused on two ion transport proteins called VCCN1 and KEA3 which play a key role in dynamically adjusting photosynthetic performance. It is known from earlier studies that VCCN1 activates sun protection if the light suddenly becomes too strong. When the light intensity decreases, the second protein KEA3 quickly breaks down the sun protection so that the plant can catch more light again. However, the two proteins VCCN1 and KEA3 have never been examined under realistic light conditions. The researchers used an innovative new approach to measure photosynthesis in combination with a targeted use of gene knockouts, i.e. plants whose genes for VCCN1 and KEA3 have been switched off. They show that the activities of the proteins VCCN1 and KEA3 depend on the light conditions the plants were raised in. Following suggestions by the head of the Plant Cultivation Infrastructure Group, Dr. Karen Cole, The researchers focused on two growth-related light factors in the analysis and were able to show that both the amount of light a plant receives, and the frequency of light fluctuations have a strong INF. The protective function of VCCN1 is only important in plants previously grown under low light. On the other hand, KEA3, which abolishes protection, was even active in high light periods when the plants were grown under conditions with elevated light intensities. Sun protection also depends on the degree of light fluctuations the plants are exposed to. When light conditions change significantly, plants produce the orange pigment zeaxanthin, which is also involved in sun protection. The production of this sunscreen is suppressed by KEA3 under high light conditions as well. Our study shows that we should not look separately at the effect of growth light and the rapid responses to light fluctuations, said study lead author Thekla von Bismarck, and added, the integration of multiple timescales and metabolic levels in an increasingly complex manner will be a major foot. This will provide key ideas to improve crop yields in the field. Make a difference. Sponsored opportunity story source. Materials provided by Max Planck Gesellschaft. Note: content may be edited for style and length. Journal reference: Thekla von Bismarck, Kubra Korkmaz, Jeremy Russ, Kira Skirk, Elias Kaiser, Viviana Correa Galvis, Jeffrey A. Cruz, DeSarah D. Strand, Karen Cole, Jürgen Eyrich, Iris Finkemeyer, Peter Johns, David M. Kramer, Ute Armbruster. Light acclimation interacts with thylakoid ion transport to govern the dynamics of photosynthesis in Arabidopsis. New Phytologist, 2022. 237, 1, 160 doi, 10. 1111, NPH. 18534. Cite this page. Max Planck Gesellschaft. Plants between light and darkness. Science Daily, the 26th of December 2022. ift.tt/m0k2 PUA. Max Planck Gesellschaft. 2022, December 26th. Plants between light and darkness. Science Daily. Retrieved December 26, 2022 from ift.tt/m0k2 PUA. Max Planck Gesellschaft. Plants between light and darkness. Science Daily. https://ift.tt/m0k2 PUA. Access December 26, 2022. Thanks for watching. Please subscribe my channel for more videos.